This protocol provides a new approach to quantifying branch dynamics of the dendritic arbor in developing neurons using live time-lapse imaging and a 3D image annotation software. This technique images and tracks the branch terminals, providing a fast and efficient method for measuring the dynamic behaviors of dendritic filopodia.
Research using this method provides insight into understanding how development and activity regulate dendrite morphogenesis. Our methods are applicable to sparsely labeled neurons in both in vitro and in vivo settings. When performing this procedure, remember that the imaging parameters must be carefully adjusted to achieve a sufficient temporal and spatial resolution.
To harvest brains from larval Drosophila, under a dissecting microscope, use one pair of No. 5 standard tip dissection forceps to hold a larval specimen in place, while using the other to carefully dissect out the brain, preserving the eye discs, brain lobes, and ventral nerve cord. Then remove the attached muscles to minimize any movement of the sample during imaging. When all of the brains have been harvested, use vacuum grease to draw a square chamber onto a glass slide, and add 20 microliters of external saline solution to the chamber. Use the forceps to transfer the brains to the chamber, and adjust the positions of the brains under the microscope with the dorsal sides facing up. Cover the chamber with a glass cover slip and press gently on the cover slip to reduce sample.